Hello everyone, I'm Ignasi Granero. I'm a predoctoral researcher at the Cellular and Molecular Mechanobiology group at IBEC. My thesis is directed by Pere Roca-Cusacs and Ion Andreu, and I'm presenting a part of it under the title of Development of a Sensor of Nuclear Force Based on Fluorescence Imaging. So we'll start by the nuclear force part, and as you know, the eukaryotic cell has the nucleus, which is an organelle that holds the DNA, and this nucleus is under forces. These forces can affect shape and volume, and can also have some downstream effects. And for example, if we compress the nucleus with an AFM tip, we have already shown in the group that the transcription factor YAP translocates to the nucleus, which can have some downstream effects such as transcription level alteration. As you can see here in the video, when we press, what we see is that the transcription factor YAP goes into the nucleus. So how does the cell nucleus sense physical changes and triggers a cell response to them? For that, we have created a sensor inspired by YAP, which means that we want it to be nuclear when the nucleus is under forces. And for that, it has a green fluorescence protein that allows us to image where the bulk of the sensor is. It has an NLS that regulates the active transport to the nucleopore, and extramolecular weight to regulate its passive transport to the nucleopore. This happens, as it's already uploaded in bioRxiv, because it works because active transport and passive transport are differently regulated when the nucleus is under forces. When we transfect some cells with our sensor and we sit them in two different conditions, soft shells and stiff shells, we can see differences. And to measure these differences, what we do is measure the nuclear to cytoplasmic ratio, which is the mean fluorescence in nucleoplasmic area divided by the mean fluorescence at the cytoplasmic area. In these two conditions, which are soft shells where the nucleus is under low forces and stiff shells where the nucleus is under high forces, we get different results. This is that we get a more nuclear concentration when the nucleus is under high forces and a lower concentration when the nucleus is under a more relaxed state. This is a very interesting result. However, so far I have only shown you data of single cell, which were done in mouse embryonic fibroblasts, as you can see here on the left side. Now we have switched to a multicellular environment with MCF10As, which are mammary gland non-tumorigenic epithelial cells. This is a very interesting system because this is a multicellular environment and these cells form cell-to-cell junctions. This is interesting because there's less clear nuclear mechanosensing mechanisms in multicellular environments, and we want to study that. For that, the first experiment we did was this monolayer experiment with this MCF10A cell line that stably expresses the sensor, but not all at the same level. So as you see here, there are cells everywhere, even if you don't see them. The aim of this was to measure the sensor ratios and see the border effect. So with this, what we did was to measure the nuclear to cytoplasmic ratio of these cells and also the distance to the edge of these cells to get the differences between the edge and the center of the monolayer. When we got the data, we got the nuclear to cytoplasmic ratio versus the distance to the edge. And as you can see here, there's a clear decrease in nuclear to cytoplasmic ratio with the distance from the edge. This is a very interesting result. However, this was done in a single confocal plane. So now, to get more data and be able to understand what's happening here, we are measuring new parameters. So we are not only measuring nuclear to cytoplasmic ratio of the sensor, but we are also measuring 3D shape of the nucleus volume, and the position in the monolayer, all this throughout time. As you can see here in this Z stack, we can see the nuclei at the left side. In the middle, we can see the sensor in green. And then at the right side, we can see them both combined. Plus, we get a bright field image. Since not all our cells are fluorescent in green, here it allows us to see the border of the monolayer and also determine which cells are touching which. So the take-home message is that we have designed a sensor of forces in the nucleus. We have tested it in single cell, and we are starting to explore the functionality of the sensor in a multicellular system to understand the nuclear mechanosensing. And with all this, I would like to thank my group, Cellular and Molecular Mechanobiology group, and also the people from the Xavi Trepats group that have helped me. And thank you all for your attention.